So today I'm going to be talking about blue-white screening. Blue-white screening is a method designed to see if your plasmid or vector has taken up recombinant DNA. I'm going to go through this kind of in a step-by-step basis, and I'll explain each step. The first step is you're going to start with your plasmid or vector. That plasmid or vector is actually going to contain a gene called LAC-Z. LAC-Z is going to code for an enzyme called beta-galactosidase. Now, beta-galactosidase is going to be very important later on when we're actually plating the bacteria because it's going to be able to actually break down xgal. Aside from our LAC-Z gene, which by the way has multiple cloning sites, aka just restriction sites, that's actually where your gene of interest is going to get inserted. So aside from just the multiple cloning sites, we also have a selectable marker with ampicillin resistance. It's also important to know that when we are plating our bacteria on our agar plate, the agar plate is going to contain not only just xgal, but it's also going to contain ampicillin. So the ampicillin resistant gene is going to allow only the bacteria that actually took up the vector or the plasmid to be able to grow. So that's kind of the first step in weeding out the bacteria that even took up the recombinant DNA in the first place. That leads me to my second step. So as you can see here, the second step would be that you are actually inserting your gene of interest into the multiple cloning site. Now what this does is it's actually going to disrupt and block off the encoding of beta-galactosidase. It's going to disrupt LAC-Z. So LAC-Z is not going to be able to produce that enzyme beta-galactosidase. And like I said, if beta-galactosidase isn't present, it's not going to be able to break down xgal. The final step is you're going to mix your bacteria, hopefully causing transformation. After this, you would plate your bacterial cells onto an agar plate containing ampicillin and xgal. The ampicillin, like I said, is going to allow only the bacteria that took up the plasmid or the vectors to grow on the plate in the first place. And then the xgal is going to determine whether or not those plasmids actually took up the gene of interest. So if they did take up the recombinant DNA, they would turn white. If they did not take up the recombinant DNA, they would turn blue. The reason behind this color is due to beta-galactosidase breaking down xgal. And so if beta-galactosidase is still present, meaning that the gene of interest didn't actually get inserted into the vector plasmid, xgal is going to kind of create that blue tint within a cell. If it turns white, then you know the gene of interest got inserted into the vector, therefore disrupting LAC-Z. Therefore, beta-galactosidase will not be able to break down xgal. Therefore, it would turn white. There would be no blue tint.